Okay, welcome back to the afternoon session of the Canola Innovation Day. Lots more great information to get through, so thank you for joining us here in person as well as for those of you that are online virtually in your home and your offices. Today, moderating our Slido questions will be Mark Smith, who's a longtime Canola Week committee member and an AAFC research scientist who studies lipids. Our first speaker, my name is Rex Newkirk. I'm a professor at the University of Saskatchewan, and I do lots of canola research. I'm an animal nutritionist, feed processing guy, so we're changing directions. We're going to look at utilization of our byproducts, particularly canola meal, and other parts. So our first speaker is Dr. Emma Stevens, a bioeconomist at the Lethbridge Research and Development Centre of Agriculture and Agri-Food Canada. And today she'll be talking to us about platforming canola value chain data to support circular economy utilization strategies. So I'll turn it over to you. Well, the presentation's already up.
Yeah, I've got a web thing, and I'll do that later. Good afternoon. Thanks so much to Canola Week for giving me a chance to present some of my research here today. I guess this is something a little different as well. I'm, as Rex mentioned, I'm a bioeconomist within Science and Technology branch at Agriculture, Agri-Food Canada. So working with many of my colleagues on a lot of different economic aspects of the scientific innovations that they are working on. But my background is part of my background is on spatial market integration and the economics of how agricultural commodity markets talk to each other and operate efficiently or inefficiently. And so just saying this is a way to explain a little bit, this is how I the perspective I'm bringing to the circular economy question, looking at it from a supply chain perspective and drawing in some of that research. Okay. Just before I get started, I wanted to acknowledge this is a project that's funded internally by Agriculture Agri-Food Canada on one of our pillars of our strategic plan on advancing the circular economy. So recognizing there is a significant amount of research support that is going to be needed to improve circularity within our agricultural sector. And I'm fortunate in this project to be working with some really great colleagues. Tim Schwinghammer, who's my co-PI on this project, is a biostatistician based out of Lethbridge. Oh, I should also, sorry Rex, I should correct. I'm actually based in the Lacombe Research Station now, just south of Edmonton. So make sure I make that correction. But a lot of my colleagues are in Lethbridge, is where I was until a couple of years ago. Dr. Roland Krobel, also at AAFC Lethbridge Research Station. Dr. Elam Rahmani, who is a data analyst who works directly with me on my team. And then, as you'll see with this data platforming project, I need a lot of help from our agri-geomatics division. So I'm working with James Ashton and Tamara Rounce, who have really been extremely instrumental in putting this data together into a great resource. And they're based in Regina. And we've also been really lucky to have some great graduate students working with us, Yi Zhang, Anushka Kumarakov, and Lena Gomez. So thanks to them. It's something that has come up in even just conversations today, so maybe this is introduction you don't need, but circular economy is just almost a mindset shift on how we're thinking about our agricultural resources and how we translate primary commodities into valued transformed commodities moving across landscapes and into different markets. The circular economy framework is thinking about transforming our current supply chain structures from a more linear approach, take, make, waste, into thinking about those resources and how we can find opportunities to recirculate them into additional value-added resources. There are a couple of different frameworks around circular economy. This one is from the Ellen MacArthur Foundation. The objectives are generally to eliminate waste and pollution. So once you have a more circular mindset, more circular structures in your value chains, the idea is things that are currently underutilized or even wasted. In the most extreme example, could be that waste could be transformed into something productive and therefore taken, minimizing the waste overall in your systems and minimizing pollution along with that, especially if you're able to take those byproducts and substitute back into your value chain for external resources that you're using right now. So that's a big idea. Circulate products and material at their highest value. So another component of it is what kind of circles you want. You want to have some sense, some information on where the relative value is in these recirculated opportunities. Part of the motivation for myself anyway for putting together this proposal is that we need a lot more data support to begin to answer those questions, to reflect that in developing circular economy. So that's kind of our big objective here, is to put this data together in a way that helps find those high value opportunities. Regenerate nature is another big component of that, with the linear pipeline, especially for natural resource sectors like agriculture, that puts pressure on our natural systems. So I think the thinking is more circular systems would alleviate some of those pressures, and we can use the excess or free up capacity to regenerate and make those resources more sustainable for the long run. This is another kind of visual of circular economy from the Ellen MacArthur Foundation, this butterfly diagram showing some very big conceptual circles. But I guess my challenge a lot with this is it's a lot of conceptual discussion about it, but how do we make it real? For us here in the agricultural sector, we're kind of thinking about this part of the butterfly diagram. How do we improve circularity in our renewable resources, like our agricultural commodities? But there's also quite a bit of research and discussion being applied to the finite materials sector on the other side of that diagram, the blue in terms of waste, recycling, and refurbishing of finite resources as well. So the circular bioeconomy in agriculture, this is getting down to what's in that circle a little bit more. You have your first pass at the primary agricultural production there, that big arrow on the top left there, going into primary processing, and then thinking about, can we transform the byproducts from that initial agricultural sector into additional resources. So you've got bio-based products, food and feed, you've got biofuels there, prolonged and shared use, so it speaks to preservation and not minimizing waste processes in the transformation, so less loss in our chain overall in terms of storage technology possibly. Energy recovery, composting. So even after all that is finished, there's still quite a bit of biofeedstock in the byproducts there. Can we get energy still from those byproducts? Can we compost them to return those resources back to agriculture? So this is also on the right-hand side. I'm trying to document some of the technologies, and some of them have come up today in some of the discussions I've been able to listen to, just improving efficiency, your resilience of your crops in terms of breeding for reducing losses at the field scale, valorization in terms of finding additional uses. So I've been able to talk with Rex about some of his work on that very big component. Precision agriculture, so it's another way to minimize your inputs. And looking at that last stage on regenerative agriculture as well. So how did we one more thing I wanted to say here. There's a number of ways you can go about making those circles. On the right-hand side is something that's often discussed, the resource cascade. So your first use for canola, and the reason I'm here is because we got into this project and discovered that the canola sector has quite a bit of really good data to build this platform. So thanks to everyone in this room for that and making our job a little bit easier. But for this cascade idea, your first use would be your oil production possibly, and your second use could be canola meal. But can you keep going? Can you keep turning those byproducts from feed production into something else? That's one way of thinking about it. On the right-hand side is recognizing the different levels of relative value. So you've got your high value at the top, possibly in your oil production versus other uses in composting or something like that would be still a use of those resources, but lower value in terms of markets. I didn't find a nice schematic for the canola sector directly, but just to make it a bit more concrete. This is from a paper by Hassan et al. On circular economy strategies for residue left over from sugarcane production, the bagasse. And they have a nice schematic there trying to contrast the linear pipeline. So this is what I've got sort of blocked off there. Just you've produced the sugarcane, there's residue and fiber left over. There's not a ton of additional uses for that waste, and it goes into landfill and produces methane and greenhouse gases. So that's like a very short, very linear take, make, waste pipeline. So they are investigating what would it mean to make it more circular. And so they take from that feedstock there, instead of just having that straight line to the waste process, they are thinking about different innovations on extracting components of that have additional value. Can they be used in bioplastics, for example? Are there other industrial uses for that feedstock? After you're done all of that, you've got a bit of that resource cascade there, so there's still going to be byproducts even if you extract maximal value, so that could be bio-waste. Can it be composted? Still nutrient value in there that might still contribute back to the agricultural sector. So this is one value chain that they're looking at the research that would be needed to make it more circular. I did find another paper by Reda et al. On biofuel and kind of circular economy strategies for the biofuel sector, not the oil seed for canola oil necessarily, but again, this resource cascade is a big component of that. They've got the primary biofuel production, but then valorization there in the middle across different components that might have additional market value that we should explore. So this is another related circular strategy. So for us, for this project, all of those conceptual ideas, I think in order to advance that being a reality, I believe we need a data platform that will help us ask those questions. As you can see with the circular economy, and my fellow presenters have had a chance to talk to them, it's not just the canola stakeholders and canola sector that's going to be involved. It could be other industries that might be interested in your feedstock for something. They need to have an easy entry into all of the information that's out there about the canola sector in order to make business decisions and think about ways that they can bring innovation to the table. So this is what we're trying to build here. Our current resources are quite scattered, so some of this is just my personal frustration with that reality. So it might just be my pet data wish, but I think we've built something that should be useful as a way to navigate through all the data that already exists about the canola sector and facilitate circular economic development. Oh my gosh, okay. So we've got 3 components here. I was looking at the wrong timer. I'm like, how do I still have 13 minutes left? Okay, we've got 3 components. One is a very overview story map, and if you've seen those from Esri, it's a nice way of presenting information to a general audience. So we have built a story map using some expertise from our geomatics division as well, talking about the circular economy and the sector. But then we have added to that significant linkages to the primary data that people can access about the canola sector. So this is public data. A lot of it links back to Statistics Canada. It's stuff that you're probably familiar with. If you look at the Outlook reports, this is the foundational information that's used to build Ag Canada's reports on the sector, but it can be used for a lot of different things. So I thought it would be helpful to have the framework and then linked with the data and where it comes from and how to get at it. And our ultimate idea is to have it updated, because those data products, like the supply and disposition table, for example, is collected 3 times a year, so you can have it be a live resource for data. We've also got a bit of a schematic here on the sector and the different components of the value chain. And again, we are building a database of data, publicly available data, to populate those boxes and have that linked in the background to the resource as well, so you can find those numbers easily. So for example, this is the production box. I'm sorry, it's a little too small to read there, but that's sort of primary production, and we've got a whole set of information at the CAR level on canola production in different regions of Canada. We also were tasked with tracking important resources within the value chain, so nitrogen and phosphorus. One idea is, can we figure out how much nitrogen is embedded within the value chain to help us figure out a way to recover it? Can we replace fertilizer with recovered nitrogen and phosphorus? But again, we need information on how much is in there in the first place in order to start to think about that. We also have quite a bit of spatial analysis. So this is a map combining our information. So for someone not familiar with the structure of our field crop value chain system, they can easily kind of see these features, but it also allows you to start to ask interesting questions about the value chain itself. So this is work. One of our grad students was a PhD in geography, and so she's come up with simple relationships to show how things are getting into the elevator and crushing system, and do some basic modeling of those relationships. And I think that's this is my favorite picture of the whole project, just because it's really bringing those nodes of the value chain together. Then we kind of followed the value chain to its end, or one of the ends anyway. So this is mapping back out livestock demand within Canada for canola meal. So it kind of is spread out in terms of primary production. It concentrates into the rail and elevator sector, and then it goes back out again. So I think that's important to document, first of all, but also it shows the challenges, I think, in creating a circle between all of those things, is that we can start to think about where does it make most sense to recirculate resources. And personally, I think that it's a hunch, but I'd like to do more research on it. That concentration in the middle of the value chain is simpler if we're looking at resource recovery. So I think that this data allows us to think about those kind of system-wide opportunities. We have developed some additional research. Now that we've got the data assembled, people want to ask interesting questions like path analysis. So my colleague is biostatistician and does a lot of work in that area, so we can take the sector and develop a schematic of how the different nodes relate to one another, and so he's used our data for that, showing, for example, the relationship between the export sector and growing degree days, for example, and we can do impulse analysis for shocks to either of those things, and how is that going to relate to the rest of the value chain, which I think would be really great. And just our future objectives, take all of this and do a mass balance in terms of looking for losses of bioresources and waste. Can we make it live in terms of pulling in the data? And can we enable more research to make things more circular? I think there's a lot of unanswered questions about our structure. So thanks for that. And we're looking for feedback. So thanks for your attention. Thanks, sorry.
Don't go too far.
Okay.
Thank you very much. Lots of things to pull together and information to gather. Any questions? Do we have any online? None online. Any questions in the audience? I have one for you. So there's been different drivers for circular economy, in different regions. I just came back from Northern Europe, and there they've done a really good job of, in my opinion anyway, of utilizing ingredients to their fullest. And part of that, I think, is coming out of, after the war, there was food security issues, there's issues, concerns. Other places, there's other drivers. What do you think in Canada could help us drive us towards a more circular economy and using our ingredients more fully? Any guesses? You're too early in your data, but I'm just wondering if you have any guesses.
I think, in Europe, it's a policy priority. So some of it is being driven by choices being made on what to support and developing that. So we're trying in SDB to support it too. So I think the sensitivity to loss of resources, if resources become more scarce, or I think trade challenges that we're having at this particular moment would make us maybe start to think about other markets, other uses, and it might be a catalyst for more circular approaches, yeah.
Awesome. Well, thank you very much. Thanks. Appreciate that. Okay, next up we have Dr. Van Toe, a research scientist at Onda on Prince Edward Island, and she's going to talk to us about evaluation of canola meal as a feed ingredient for Atlantic salmon.
Thank you, Dr. Resch, for the introduction, and good afternoon, everyone. My name is Van. I am a Vietnamese. I am 100% made in Vietnam product. But no worry, I'm not going to talk about myself today, but I will talk about canola meal, 100% made in Canada product. So I work for Onda. So Onda is a research contract organization focusing on aquaculture research. So today I will present some of our research activity and results we have done, focusing on exploring canola meal as a protein ingredient in salmon feed. So regardless of where salmon is produced, in Canada, Scotland, or anywhere, feed is always the biggest component in production cost. And feed is also the biggest contributor into the greenhouse gas emissions. So aquaculture production is anticipated to increase in general, and aquaculture and salmon aquaculture production is also increased. We see that the feed ingredient will increase. For feed and their 4 ingredients to be effective, there are some important factors that we always consider, including the ability to support the animal genetic growth potential, ability to deliver cost-effective performance, and the ability to support the sustainability goals. So sustainability has moved from the minor topic to the central topic right now in aquaculture. We can see the number of publications increase significantly in recent years. So sustainability and economic factors influence how we select and formulate diets. So have a look on the Norwegian salmon feed. We can see that the non-sustainable limited ingredient and expensive ingredient like fish meal and fish oil, the inclusion level significantly reduced in the past 3 decades, whereas we can see that plant-based ingredients has been included at higher and higher inclusion level. When we look at in 2020, 73% of ingredients in Norwegian salmon feed came from plant, and plant protein is the biggest component in this chart. Among the protein ingredient, we see the soy protein concentrate is the most dominant ingredient. So to reduce reliance on soys and to diversify the protein ingredient, we can see that canola meal is one of the promising ingredients, but it has been underutilized. So canola meal is the byproduct of the canola oil industry. So canola is the second largest oilseed crop. So it provides an excellent scalability supply and enable sustainable circular economy efforts. And in fact, canola is a vegetable ingredient, therefore it will face less restriction compared to some other ingredient, for example, like poultry byproduct meal in Europe. With 41% of protein and reasonable amino acid profile, and the cost is less than $500 per metric ton, canola meal offers both nutritional and economic benefits that can be a practical option in feed formulation. Therefore, the objective of our research is to evaluate canola meal as the protein ingredient in salmon feed. So there was 4 main research activities. First, measure the digestibility of canola meal. Second, evaluated the effect of canola meal inclusion level in the diet on the performance in terms of the load and feed utilization. And look into the environmental impacts of canola meal in salmon feed using life cycle assessment and explore potential health benefits when the fish fed with the canola meal. So the first study is the digestibility study. We have 1 reference diet and 1 test diet that was created by combining 70% of reference diet and 30% of canola meal, which is one of the traditional methods of evaluating the digestibility study. We had 3 replicate tanks per diet, 30 fish per tank, and the fish size was 50 grams. The study was run for 62 days, long enough to acclimate the fish to the water environment as well as to collect enough fecal material to analyze the digestibility. And here's the result. We can see that the apparent digestibility coefficient of protein and essential amino acids in canola meal range between 73 to 88%. So consider this is 41% of protein. When we compare with some other ingredients that typically has higher protein and more expensive, we can see that canola meal has equal or even better the digestibility than these ingredients. The second study, we look at the growth performance. So in this study, we have 4 diets, 0, 5, 10, and 15% of canola meal. The diets were produced in-house. In our company, we have the pilot scale extruder where we can make a diet in-house. So all of the diets have equal percent of protein, 45 to 46% of protein, to equal percent of lipid. And when we talk about protein ingredient, not only about nutrition, but we also have to think about whether the ingredient can be incorporated into the compound feed or not. So with and our company, we see that canola meal didn't have any negative impact on the pellet quality, including the bone density or sinking velocity of the pellet. So we used the pellet for the feeding trial. We had 3 tanks per diet, 50 fish per tank, and the fish size was 245 grams to begin. We hand-fed the fish to appearance satiation 3 times per day, and the feeding trial was 103 days. And here's the result. So in terms of feed intake, we don't see any significant difference between treatments that indicated that canola didn't impact the palatability of the diet. For the weight gain and FCR at 15% inclusion level, we see that significant reduction compared to the control. However, at 5 and 10% inclusion level, there's no significant difference. We analyzed the data further using broken line analysis, and we identify about 7% of canola meal is the optimal inclusion level based on the weight gain and FCR. So whether feed canola impact the fish health of the fish or not, when we look at blood biochemistry, we don't see any significant impact of the canola on the immunity response or antioxidant parameter. We also look at the liver histology, and all of the fish has similar morphological performance. So gut histology is one of the main parameter that people look at the fish health. So we look at submucosal thickness and villi length of the gut. There was some variation, but technically there was no significant difference. When we look at gut inflammation, we can see that the fish fed with canola meal have a tendency to have more inflammation compared to the control. However, this difference was not statistically significant, and is also not necessary to translate into the negative impact of the growth performance of the fish, especially up to 5 or 10% inclusion level, as I just presented in the previous slide. So feed cost, we see that at 10% inclusion of canola meal give us cheaper cost in the feed compared to the control. So when we look at whether the fish-fed canola meal has any environmental impact or not, we use the life cycle assessment, and we run the model for both sea cage and indoor recirculating aquaculture system. Although not significant, but we can see that there's some positive impact on the environment, including the CO2 emissions, water use, and land use. So to explore, is there any potential health benefits of canola meal on the fish health or not, we screened some pathogens, the common pathogen that cause the disease in fish. So there was 9 pathogens that we're looking for, and we see that the food extract of canola meal can reduce the growth of Tennessee baculum fimacansis. So Tennessee baculum fimacansis is the bacteria that can cause the mouth rot disease, and this can cause significant economic loss due to mortality. Currently, there's no available vaccine, and the antibiotic use can be very expensive. So from these data in vitro, we performed an in vivo study to see whether fish-fed canola meal can have better survival or not. So we conducted an in vivo challenge study where the fish fed 2 diets, zero control diet, no canola meal, and the diet with 15% of canola meal. So the fish, we had 3 replicate tanks per diet, 103 fish per tank, and the fish size was 45 grams. The fish were fed for 35 days before they got being challenged with the bacteria, and then we continue feed the fish with this diet and monitor the mortality. And here the result, we see the significant improvement in the survival, up to 53% improvement in survival relative to the control. So in conclusion, we see that inclusion of canola meal in Atlantic salmon diet can provide digestible protein as essential amino acid, can maintain comparable growth and feed efficiency, can support the normal health when we look at the blood, liver, and intestine health. It also shows resilience against Tennessee fimacansis and offer environmental advantages. So when we talk about sustainable aquafeed, we tend to focus on planet. Planet is absolutely important, but equally important, a good ingredient is the one that can work well in the compound pellet. It has to be able to support the raw performance and health of the fish, as well as to have affordable price to make economic sense. Therefore, sustainability requires a holistic approach that we need to consider environmental, nutritional, economic, and functional factors that allow us to deliver meaningful results. So with today's result, we can see that canola meal has satisfied many of these ingredients, and therefore it has been demonstrated canola meal can be one of the candidates to be used in salmon feed up to the level less than 10%. With that, I want to thank you, Canola Council, for sponsoring all of the project, and I thank you everyone for your attention. I'm looking forward to have any easy questions.
If they give you easy questions, I'll give you hard ones. Hey, questions? Mark, you have one?
We do, so thank you, Van. We'll start with an easy one. Was the meal cold pressed, or is this solvent extracted meal that we're using in this study?
Can you repeat the question?
Was the meal solvent extracted or cold pressed that was used in the study?
Solvent extract.
Okay. And the second one was for marketing. Would you be required to use non-GM canola? Do you think that would matter to the fish food market?
In Canada, we can see that GM is very common. In Europe, it's a bit more tricky. However, it's a harder market. However, I think that there's some more openness for the GM product nowadays. We can see even some European country now allow the GM oil, canola oil. So that means that I think there's the potential over there for GM canola meal.
Good.
We do have one more online, unless anyone on the floor has a question. So the last one was containing glucosinolates. So do you know anything about the glucosinolates in the meal and if they had effect on the health of the fish?
I think this is a question for Curtis. No, I don't think so. Canola meal, genetically, we have been bred with low uric acids and glucosinolates, so the level is much lower to be qualified for use right now. So, and glucosinolate is hypothesized to influence like thyroid hormone, and we analyze the blood biochemistry, and we don't see any significant different impact at all.
Thank you.
Excellent. Questions from the floor? I've got one for you. So one of the unique things about canola meal compared to the other, you showed corn gluten meal and soybean meal and soy concentrate and things, is that canola meal's got a higher level of fiber, more lignified fiber. Do you think it's possible that fiber might be part of the response, might be responsible for some of that ability to fight off some of the disease? What role do you think the fiber might be playing in there?
This is something, that's a good question too. So we are now looking into some literature, and it looks like some bioactive compound of canola, like phenolic acid, that have been reported to have antimicrobial activity. That's why we want to look in further into the mechanism whether phenolic acid can be the candidate to reduce the disease incidence or not.
Right. Good. Any other questions? Good. Well, thank you very much. Appreciate that. Appreciate you coming all the way here for the presentation. And Dr. Van To's very committed because tomorrow morning at 6:30 I'm picking her up and we're driving North Battleford to see my research facility. So she's going to see Saskatchewan in its fullest. Next up we have Dr. Tofuko Woyengo, an associate professor in the Department of Animal and Veterinary Sciences at Aarhus University in Denmark. And he's going to talk to us about growth performance and gut health of nursery pigs fed canola meal based diets. And we're just going to get that up online here in a second.
Hello, how are you?
I'm good. How about you?
I'm fine. Yeah, you can hear me?
We can.
Okay, yeah, so I'll share the screen and start, yes.
Okay, we see you. Now we're just waiting for your presentation to come up.
Okay, can you see the presentation now?
Is the presentation up there? I can't see it. No, not yet.
Not yet?
Just 1 second. Okay, now we're good. Go for it.
Okay, yes, yeah. Okay, yeah, so I think I can start, yeah. So thanks a lot for the introduction. My presentation is going to be on growth performance and indicators of gut health of weaned pigs fed diets containing canola meal. So I go straight to the introduction. It is well known that post-weaning diarrhea leads to reduced feed efficiency and increased mortality, leading to huge economic losses in swine industry. So that is a fact that is well known. And it's also well known that the antibiotics, like the addition of antibiotics in darts for the weaned pigs can result in reduced post-weaning diarrhea. But the problem is their use is being discouraged currently because they are believed to lead to development of antimicrobial resistance. So based on this, then there is a need to develop alternative strategies for managing the post-weaning diarrhea of the pigs. So in order to come up with alternative strategies, we first need to understand or know the conditions that leads to post-weaning diarrhea. So we need to understand that before we can come up with any meaningful strategy. And if you look at this slide, that first picture on the left, it is of the gut mucosa, that is the small intestine. And if you look at it, you can see it is lined by those special cells known as epithelial cells, which are joined together with a special type of proteins known as the tight junction proteins, like the reddish and the bluish colored proteins that join those cells together. So if a pig is healthy or an animal is healthy, like it has a healthy gut, those tight junction proteins prevent the flow of pathogens or toxins within the mucosa and hence within the body. However, any form of stress, such as post-weaning stress, can lead to oxidative stress, and oxidative stress can lead to generation of free radicals, which can then destroy the tight junction proteins, leading to the translocation of toxins and pathogens within the mucosa, thereby injuring the mucosa and leading to increased susceptibility of the pigs to infection. So that is what results in post-weaning diarrhea when we have that increased susceptibility to infection. So just based on what we have covered, for a product to be effective in managing post-weaning diarrhea, then it has to either reduce the growth of pathogenic microorganisms within the gut lumen, or it has to reduce oxidative stress and therefore the destruction of these tight junction proteins, and therefore the translocation of toxins to the body, or be able to do both. If it is the product is able to do both, like reduce the growth of pathogenic microorganism and also reduce oxidative stress and therefore the destruction of the tight junction proteins, then that will be the best. So canola meal, we all know that it is a good source of amino acids, and therefore it is included in diets for pigs to supply amino acids. But there are extra good things about canola meal, potentially good things about canola meal. One of them, it's because it contains insoluble fiber, and another one is because it also contains some low levels of glucosinolates. So the insoluble fiber is good, can be good for weaned pigs because it can lead to reduced growth of pathogenic microorganisms in the gut by increasing the digester flow rate in the small intestine. So that slight increase in the flow rate of the digester can minimize time, like reduce the time for the microorganism to interact with the mucosa and attach to them. So that is potentially one way in which canola milk can reduce E. coli infection. Another one is the glucosinolates have been shown to have antimicrobial and antioxidant activities. That means that the glucosinolates can also reduce the growth of pathogenic microorganisms within the gut and also reduce gut permeability by reducing the oxidative stress. So those are the good things that can make canola meal to be potentially a functional feedstuff. But it's also known that canola meal, the glucosinolates in canola meal are bitter and therefore they can reduce feed intake. it's also well known that the glucosinolates can interfere with the thyroid and liver functions because they have to be detoxified within the liver, and also they result in a reduced uptake of iron by the thyroid gland for the synthesis of thyroid hormones. So based on this, then there's a need to identify the optimal level of canola meal in the diets for weaned pigs that can improve the gut health, but at the same time should not compromise the performance of these weaned pigs. So that was the main aim of this study. So then the objective was to determine the effect of inclusion of canola meal in diets for weaned pigs on growth performance and indicators of gut health. To achieve that objective, two studies were conducted, and the first one was to determine the effect of increasing the level of canola meal in diets for weaned pigs on growth performance and indicators of gut health, with the goal of identifying the optimal level of the canola meal in the diet. So this study was conducted already, and it has been published in the Journal of Animal Science. This table, the first table up there, shows the ingredient and nutrient composition of the diets that were fed in that study. And as you can see from it, the diets were based on the corn and soybean meal. And that diet, like the basal diet, was without or with the increasing level of canola meal. That is actually 5 levels of canola meal, 0, 10, 20, 30, and 40%. The diets were formulated to similar net energy values and also to similar standardized digestible amino acid contents, represented here by standardized digestible lysine content, but the rest of the amino acids were balanced also, and also the calcium and phosphorus. The diets were fed for 6 weeks, that is 42 days, and they were fed in 3 phases. That is phase 1 for the first 1 week, phase 2 for the next 2 weeks, and then phase 3 for the last 3 weeks. The table below there is of the chemical composition of the canola meal used in this study. And if you look at it carefully, you'll see that the crude protein, the fat, and glucosinolate content in this canola meal used in this study, those values are similar to the values that have been previously reported for the conventional solvent-extracted canola meal, that is the Brassica nepus. So it was a representative of the canola meal that is used in the pig industry, or both pig and poultry industry. So moving to the results from this first study, this figure here shows the effect of canola meal on growth performance, serum IgA concentration, and the urine lactulose to mannitol ratio of the pigs. So the first figure is of the effect of canola meal on the overall, like the entire study average daily gain. The second one is of the effect of the dietary canola meal on IgA concentration. IgA stands for immunoglobulin M concentration at the end of phase 2 feeding. And then the third figure is for the lactulose to mannitol ratio in the urine of the pigs at the end of the second phase of feeding. So the IgA level reflects immune response. That is, an increase in the level of IgA in the blood indicate an increase in immune response, and therefore infection. Whereas the lactulose to mannitol ratio here indicates gut permeability. That is, the higher the ratio of the lactulose to mannitol, the higher the permeability of the gut, especially the small intestine. So if you look at each individual figure, to start with the one for the performance, we can see that an increase in dietary level of canola meal from 0 to 20% resulted, like when it partially replaced the maize, which is corn in US, corn and the soybean meal, it resulted in an increase in growth performance up to that level, like 20%. It resulted in an increase in growth performance. However, a further increase in the level of canola meal in the diet from 20 to 40% resulted in the reduction in the growth performance of the pigs. So then we ended up having a quadratic response, like an increase and then a decline. But what I'd like you to note from this is that the growth performance of pigs fed the 40%, the diet containing 40% canola meal did not differ from that of the pigs fed the diet with the 0% canola meal. That is the diet based on corn and soybean meal. If we move to the second, and before we move to that, the average daily feed intake followed the same trend as the average daily gain. So we also saw a quadratic response for the average daily feed intake, although I have not presented the figure here. And because of that, there was no effect on the feed efficiency because it remained the same, because the feed intake and the average daily gain increased with the level up to 20% and then decreased with the level up to 40%. So I have not presented that, but the trend was the same. So now if we move to the second figure, we can see that the trend was quadratic, but in the opposite direction. That is, an increase in the dietary level of canola meal from 0 to 20% resulted in a reduction in IGA production, but a further increase in the level of canola meal in the diet from 20% to 40% resulted in an increase in IGA level in the blood. If we move to the third figure, you can see that an increase in dietary level of canola meal from 0 to 40% resulted in a linear increase in lactulose to mannitol ratio, implying that it increased gut permeability. So because it's known that an increase in the level of IgA in the blood reflects the immune response, and an increase in immune response reflect an increase in infection, which is not good for growth performance, then we can say that this increase in average daily gain of pigs in figure 1 from when the canola meal was increased from 0 to 20% in the diet can be attributed to the reduction in IgA level and therefore immune response. IgA level when the dietary level of protein was increased from 0 to 20. And this reduction in average daily gain in figure 1, when the dietary canola meal was increased from 20 to 40%, can be explained by the increase in the level of the IgA in the serum due to the increase in dietary level of canola meal from 20 to 40%. Also, that reduction can be explained by the increase in gut permeability when the level of canola meal was increased from 20 to 40%. You can see, although it is linear, but the increase in lactulose to mannitol ratio was so huge when the level of canola meal was increased from 20 to 30 or from 20 to 40. So those values for 30 and 40 appears to be higher compared to the levels of 0, 10, and 20. So that can also explain why there was a reduction in growth performance of pigs in figure 1 when the level of canola meal was increased from 20 to 40%. So it is surprising that this happened, like there was an increase in gut permeability in the last figure when the canola meal was increased from 0 to 40%, because we had hypothesized that canola meal is going to reduce the gut permeability because of the presence of glucosinolates and also the insoluble fiber. However, it should be noted that although glucosinolates are detoxified within the liver, the detoxification actually starts within the mucosa. So we hypothesize that the glucosinolates were detoxified within the intestinal mucosa, leading to increased metabolic activity within the mucosa and therefore oxidative stress when a high level of canola meal was fed. So it is just an hypothesis, yeah, but there is no clear reason. And also, though I have not presented here values about liver and thyroid functions, there was also a linear increase in liver size and also an increase in thyroid gland with the increase in the dietary level of canola milk. And also the thyroid hormone level was reduced with the increase in the dietary level of canola milk. So I don't have those slides, but I'd like you to note that. Yeah. So in summary, from that study, we can see that the dietary canola meal at 20% improved growth performance and reduced IgA production within the body, which means it reduced the immune response. Also from that study, we can see that the dietary canola meal at 40% did not affect growth performance, but increased gut permeability. So based on the results from that first study, it was established that canola meal at 20% is optimal for improving performance and reducing immune response and therefore gut infection. So based on those results, we conducted the second study to determine the effect of feeding E. coli challenge pigs with the diets that contain canola meal at 20% on growth performance and indicators of gut health. And the reason why we had to challenge the pigs with E. coli is because the results from E. coli challenge pigs are the ones that are more applicable to pigs that are raised under commercial conditions. So in this study, 3 diets were fed. The diets were controlled diets similar to the one fed in the first study, exactly the same diet, without or with antibiotics, or canola meal at 20%. The diets were fed for 3 weeks, that is 21 days in 2 phases, just like in the first study. Phase 1 was fed for the first week and phase 2 during the last 2 weeks. And the pigs were challenged with E. coli at the end of phase 1. So moving to the results, these 2 figures here shows the effect of dietary antibiotics and canola meal on growth performance. That is the overall average daily gain at day 21, and also the white blood cell count at day 21 when at the end of the study. And from it, you can see that dietary inclusion of antibiotics improves the overall growth performance of those E. coli challenge pigs. Also, dietary inclusion of canola meal improved the overall daily gain or performance of those pigs, but the performance did not reach that of the diet containing antibiotics, which means both of them were effective, but antibiotics were more effective than canola meal. Sorry. And if you look at the second figure, you can see that addition of antibiotics in the diet reduced the white blood cell count significantly, and also the inclusion of canola meal in the diet tended to reduce the white blood cell count. So it was not so as effective as antibiotics in doing that. But because white blood cells also reflect immune response, the increase in growth performance due to dietary antibiotics and canola meal can also partially be explained by the reduction in white blood cell due to the inclusion of the same products, that is antibiotics and canola meal. So in summary, we can say that the dietary canola meal at 20% improved growth performance and reduced white blood cell count of E. coli challenge pigs, which means the improved performance was partially due to reduced immune response and therefore due to reduced infection. So overall, we can conclude that the dietary canola meal at 20% can improve growth performance of weaned pigs and reduce immune response, and canola meal can therefore be used as a functional feedstuffs to manage post-weaning diarrhea in weaned pigs. So finally, I'd like to thank the sponsors of this project. It was the Agriculture and Agri-Food Canada, Canola Council, Alberta, Manitoba, and Saskatchewan, Canola Growers Associations, and also South Dakota State University, where this research was done. With this, I'd like to thank you and I'll be happy to answer any questions that you may have.
Excellent. Thank you very much. Do we have any questions online, Mark?
Not yet.
Okay, any questions from the floor? I've got one for you. So the canola meal seemed to help fight against the E. coli infection.
Yes, yes.
But it didn't respond as well as antibiotics. Is there any single, like, would we expect a single alternative to antibiotics to work as well as an antibiotic? Like, it wouldn't be expected that canola meal would be able to do as well as an antibiotic on its own, would it?
Yes, ideally that is what we would want, but it appears it is not possible. So it is not possible just to have a single strategy, but it is good to know that it has some positive effect so that we can see if there are other strategies that can be combined with the canola so that they can be as effective as antibiotics. That is what I can say. But the good thing that we know that it has some effect, it has some positive effect.
And I guess until recently, we didn't recommend using canola meal in a nursery or a starter diet in baby pigs because of the fiber. But now I assume it's the fiber probably that's causing, that maybe having this positive effect. So do you think people will start using more canola meal in nursery and starter diets?
Yes, I think so. I think people should start using a lot of it because other studies also done in Spain have shown that insoluble fiber, like from hot hulls, can have positive effect on, or for like a coarse ground wheat, can have positive effect on the gut health of wild pigs. So it is something that people can consider. And also, there is a need, like if we have money, there is a need, we can fractionate this canola and establish if it is actually fiber or glucosinolates that are more effective. But for now, I think with the results that we have, it will be beneficial to be adding canola meal, say at 20% to improve performance. Yes.
Excellent. I've got a question on the floor.
Yeah, thanks for your presentation. I'm wondering about palatability, and did you find any difference in the way the pigs, like was it palatable? Did they enjoy eating it? Was there any, like in those feeding trials, was there any signs of them like trying to push and get away from the canola meal and eat the other feed in the ration? Or can you comment on palatability in general at 20%?
Yeah, so we didn't, I think you are talking about preference. There is a different feed preference versus say voluntary intake. I don't know, I can explain it, but we didn't do that. Preference studies where like you have 3 feeders within the pen, and you watch like pigs have a freedom to either eat soybean meal based diet or canola meal based diet, and then you see them either preferring one or not. So we didn't do that. But if you don't do that and you just feed canola meal, they are going to eat it anyway. And it is difficult to know whether they'll prefer it or soybean meal. But what we observed in this study was that quadratic response to the dietary level of canola meal regarding our daily feed intake, like an increase to 20% resulted in an increase in feed intake, but a further increase to 40% resulted in reduced feed intake. Yeah, so we can say based on that, if pigs are not given canola meal, a diet containing canola meal alone, they may prefer the soybean-based diet, meat-based diet.
Excellent. Well, thank you very much, and thank you for making this presentation for us today. We'll appreciate it very much.
Okay, thanks for giving me the opportunity. Yes, thanks.
Great. And next up, we have Saluja Karki. She's a master's student in the Department of Animal and Poultry Science at the University of Saskatchewan. She's actually one of my graduate students, so I'm very pleased to have you come and present to us today.
Thank you. Good afternoon, everyone. We talk a lot about, let's go back. So when we talk about canola meal, we talk about it being a protein source. And today, I'm trying to go beyond protein and talk about micronutrient, especially choline, and know how the processing done for the canola meal production impact its bioavailability and how well chickens could utilize it. So as I mentioned, when in livestock industry, when we talk about canola meal, it is a source of protein. But then when we look at the overall nutritional profile, canola meal is also a significant source of micronutrient, especially choline. And chickens don't just grow on proteins. They need their fair share of micronutrients like we humans do. But there is very little information on the micronutrients, especially choline bioavailability. And very important thing is we should know why this is important. Choline is a micronutrient which does a multiple function at the same time. It is a part of a cell membrane as phosphatidylcholine. It also is a part of nerve signaling as acetylcholine. And it helps in fat metabolism in liver and is also act as a carbon methyl donor. It is a very major, it is not a minor nutrient, sorry, my bad. It is not a minor nutrient as it deficiency shows. And in the mid-20th century, we got to know that there is a choline deficiency, and it was shown by when birds started showing cirrhosis, which you see in the bottom where they have a slip tendon. It also causes the fatty liver. When there is an inefficient utilization of a fat, the fat start depositing in the liver, and there it causes the fatty liver. With all of these, it reduces the performance, it reduces the growth performance of chickens. So to prevent this, we have a requirement for choline by NRC, and that is 1,300 mg per kg for starter and 1,000 mg per kg of feed for grower. Also, we do know that choline in canola meal is poorly available, but though there has not been much research done in 1997 shows a poor bioavailability, but they don't explain the type of meal they use, as there are two different kinds of canola meal available in a market. One is solvent-extracted canola meal, and the other one is expelled, which is used in animal feeds. In these two different types of meal, most of the preliminary processes are kind of similar, but it differs in the way we extract oil. In expelled canola meal, we use expeller press, where the temperature goes up to 140 degrees centigrade for less than a minute to extract the oil, whereas in solvent-extracted canola meal, we use the solvent, organic solvent, that is hexane, to extract the oil, and further on the meal to evaporate the excessive solvent, it goes through a process called desolventization and toasting. In this, the temperature goes up to 110 degrees centigrade for about 45 minutes. So we hypothesize the changes and the differences in the time and the temperature might have an impact on the choline bioavailability in chickens. To add a complexity, choline in canola meal exists in the form of synepine. It is an ester form. So when chickens eat canola meal, the choline in canola meal is not readily available to them, as it goes into the hindgut and it breaks down and releases choline. This choline is further metabolized into trimethylamine oxide, which used to be a problem, fishy odor in a brown-celled eggs previously due to that, because chickens lack the enzyme to convert it into odorless form of trimethylamine oxide. So overall, our problem statement is that there is a very poor understanding in the bioavailability of canola meal across different types of canola meal available in the market. And our research question is, how does this different processing method, the expelled versus solvent extracted, impacts the bioavailability of choline in these broiler diets? And for that, we conducted a 26-day-long feeding trial where we had 2 different kinds of canola meal in 3 different inclusion levels of 5, 10, and 15%, which all were prepared in Canadian Feed Research Center in North Battleford, and we conducted our trial in the Poultry Research Center. where we looked at, where we housed 5 birds in bioassay cases, and we had 8 replicates. In that, we looked at the weekly growth performance, and we also collected the fecal samples at the end of the trial, and at the end, we euthanized all the birds, and then we collected the digesta samples. Those digesta samples were looked at the choline and the synapine using liquid chromatography, and all the data were analyzed using SAS, and here are the results. For the growth performance, there was no significant difference in terms of the average weight gain and overall average weight gain and the feed conversion ratio. The only significant difference was in the feed intake, where the highest feed intake was with 15% canola, 15% of ECM diet. And looking at the energy and digestibility, as we know that expelled canola meal has higher residual oil in it, which it shows over here, and the highest energy metabolized was with 10%, which was with the moderate inclusion, but as we increase the inclusion levels, the fiber dilutes it, and then there was it decreased in the 15% inclusion of expelled canola meal. However, there was much not difference in the solvent extracted meal. Now, moving towards the slide, which is very important for us, is that we found that for these synapine content, it was 1/3 of what we have been seeing in canola meal. Previously, it's over decades ago, I think with the research, it says that there is 1% synapine in a canola meal, but it has gone down to 0.67%, which was kind of similar in both types of meal, which might be due to the breeding in canola meal. And the other thing to note over here is that since both the synapine content is kind of similar in both, it's the choline content that has gone down significantly in the solvent extracted canola meal. Both the meal kind of have a similar processing method other than how the oil extracted, the difference I had talked about earlier. Though we don't know exactly why the difference is, but we speculate that the desolventization and toasting method, where it uses the temperature for about 45 minutes, might have degraded the choline. Now, going towards our slide from the ileal choline digestibility, where we see that the digestibility of choline from the expelled meal was higher, and it has an increasing trend with increasing inclusion levels, whereas for the solvent extracted meal, the choline digestibility goes down, and it kind of follows the linear trend. Yeah. And we also looked at the synapine digestibility in ileum, which shows that there was the highest, lowest inclusion level had the highest synapine digestibility. It kind of shows that chickens might have limited synapine esterase activity, and they could digest it, but as you increase the inclusion level of canola meal, they couldn't digest it. So there is a decreasing, you could see a decreasing trend in both the digestibility of synapine of both kind of meals, but still expelled canola meal has higher synapine digestibility. So to further discuss is that looking at the growth performance data, we can say that chickens compensate for the lower energy by adjusting their feed intake, as the highest feed intake was with 15% solvent extracted canola meal. And we saw that expelled meat at 10% inclusion levels have both higher energy and also the higher choline content, which could be beneficial for the producers. We also saw we have seen a limited synapine esterase activity in the chickens, as they could digest some of the synapine in the lowest inclusion levels, but the activity gets saturated as the concentration of the meal increases in the diet. So to further conclude of what we have seen so far is that synapine is 1/3 of what it was previously thought of, and expelled canola meal is a better source of digestible choline. Also, the digestibility ranged from 45% to 76%, and the efficacy of choline digestibility or utilization differs basically in the meal that you use. And like lysine used to be impacted by the toasting method previously, there might be some complexes being formed in this during the process of desolventization or toasting, which might be making the choline less available to the chickens. And with that, we have seen that more than 50% of choline is available. As in the current trend, we use choline chloride to make up to the requirement, to match up the requirement of birds in a general practice. And there is also a very hygroscopic compound, and it's increasing a lot for a producer. But if we could partially fulfill the requirement of choline with using a canola meal, it would save a huge chunk of money to the producers, and also it would provide a missing piece of puzzle to the canola meal overall nutritional profile. And in further, we are looking to improve the bioavailability of choline by using enzymes like synapine esterase or any other kind of carbohydrates, which is generally added in our diet. That is our further research. And at the end, I would like to thank the funders, Sask Oilseeds and Saskatchewan Ministry of Agriculture. I would also like to thank Structural Science Center, where I did all the analysis, and Poultry Center. I would also like to thank Canola Week for giving me this opportunity, and my supervisor, Dr. Rex Newkirk, for giving me this opportunity and giving me the whole chance to do it. Thank you.
Questions? Yes, go for it.
Thank you, very nice talk. I just wondered, did you actually look for different forms of choline in the meal? Because you can certainly imagine that there'll be a difference between cold pressed versus solvent extracted if you're looking at phospholipids.
Yeah, that was a very good point. We did not look specifically at different types of choline. We just looked at choline, the synapine, choline, and its metabolite. That would have been a great part, maybe in future.
So that may explain some of the differences.
Yeah, thank you.
No? Any other questions from the floor? So right now when we formulate diets, or most nutritionists, we don't consider any of the choline to be bioavailable. Is this an opportunity for us to maybe start considering the choline that's in the ingredients? And you're a nutritionist now. You're an actual nutritionist. Will you start considering that?
I think as a nutritionist, we would like to have a value. Like from this research, we said it's 50% 50% available. So if we could do more research in future and have a value, I would be really happy. I would really love to get a choline from canola meal, which is a natural source, rather than supplementing it from choline chloride. Definitely in future.
Thank you. Good. Okay, well, thank you very much, and thank you for participating in our session on canola utilization. And let's take a break until 2:40. Thank you.